Got awfully quiet there. Okay, I think we'll get going. I know many of you, but some of you may not know me. My name is John Kress. I'm a curator here in the Department of Botany and also the interim Undersecretary for Science at the institution. And I've been helping push along this effort we're now calling the Institute for Biodiversity Genomics at the Smithsonian. And it's good to see you all here, ready to hear about it. A lot is going on with the Institute for Biodiversity Genomics. Please come on in. We just thought we would keep on time here, or Lee is going to kick me if I don't do this quickly. The Institute for Biodiversity Genomics is moving ahead in a big way. We started ourselves off last December 12th with that issue on avian genomics that appeared in Science magazine. The number of our scientists were involved in. It was a good start for this effort. This recent cycle, and we'll hear from half of the 17 awardees who received the initial awards from our dual granting program between the Institute and the Global Genome Initiative. We were able to fund 17 projects at over $360,000. That's a good start for us to get going. Most of these are grants, are seed grants to look or get initial data that will allow you to go on to the next step in your genomics projects, and that's what they're really about. We will have another competition next year, so be ready for that, those of you that did not receive awards this year. Next year, we're going to be probably emphasizing teams of researchers that are coming together to pursue particular genomics projects. Besides this award cycle, we also have a number of biogenomics fellowships now. We're hoping to increase that. Obviously, a lot of you that have Buck fellowships or other fellowships are doing genomics, but we're trying to have a certain number of dedicated biogenomics fellowships as well to make sure we keep that moving forward. You should also know that the Smithsonian and the Institute for Biodiversity Genomics realize that we can't do everything ourselves here at the Smithsonian, and so we're moving forward on a number of partnerships with external organizations such as Oak Ridge National Lab, the Institute for Genomic Biology at the University of Illinois, BGI from China. Some of you may have been involved in that visit. This is all to expand our ability to do more and more in biodiversity genomics, and that's what the whole point is, and you'll be hearing more about those. We also are moving forward. Obviously, we can't do this without increased funding. We are now have a increase in the federal budget for biodiversity genomics. Of course, that depends on a lot of things that happens on Capitol Hill. But we also are making a major effort in terms of external philanthropic fundraising. And those of you that haven't seen the case statement for biodiversity genomics, I have a few copies here. But this is what Jenny McMillan and myself and a few others, along with the development offices at the various units that are involved in genomics, are banding together to actually find us external funds to make genomics happen. Before I introduce Lee, who will Warren, who will kick this off, I just wanted to thank both the executive committee, and many of them are here. There's about a half dozen, maybe 8 people that represent all the science units or the biodiversity units at the Smithsonian that meet on a bi-weekly basis to push biogenomics forward. And there's also an external, or there's a wider advisory committee, and again, some of those are here, that help us determine what direction biogenomics is going. So that's a big pan-institutional effort, which I think is very important. All right, enough of that background. Warren Johnson, let's start things off.
Thank you, John. It's a real pleasure. As people come in, go ahead and find seats. If those of you, if you have an empty seat in the middle, if you could squeeze in on either side, that would help us. We are trying to keep a pretty tight schedule, so we will hold questions till the end for everybody as we go forward, and I will just give brief and short introductions as we go along. So our first speaker, Karen Osborne from the Natural History Museum, is going to present her project, A Comparison of Vision Genes Across the Hyperid Amphipod Tree based on collections in the Gulf of California, Mexico.
Thank you, Warren. Can we have the slides? There we go. Okay. Thank you. I'd like to start by acknowledging my co-investigators in this project, Bill Brown from the University of Miami and Jamie Baldwin-Fergus. was recently a Peter Buck postdoctoral fellow here at the museum. This project was funded to look at transcriptomes of hyperids. Hyperids are amphipod crustaceans. They are a relatively diverse group, about 340 described species. Unlike most of the projects that I work on, we actually have a relatively robust phylogeny to start with to kind of guide our taxon selection with this project. The really interesting thing about hyperids is that they have this amazing variety of eyes. They have everything from completely absent eyes, there's no structure there for them whatsoever, and they have no ability to see, to things with minute pigment cups that are so small you can't even see them on this picture. There's things with very large eyes, things that have crystalline cones connecting their lenses to their retinas. There are animals that take their single eyes and split them into two eyes so that they can see in multiple directions at the same time. And in the example that I'm showing you here, they actually use their exoskeleton as blinders to direct the light into the eye from different directions. There's animal groups that have actually duplicated their eyes, so they have two pairs of eyes. Typically, the eyes look in opposite directions. In Phronema, this example here, they also have extremely long crystalline cones in in one pair of the eyes. There's animals that have sheet-like retinas, animals that have multiple retinas within a single eye, animals that have tubular retinas, and so their eyes are cylindrical. They can see in 360 degrees around their head. Doesn't do them much good for what's ahead of them, but all the way around, they're doing good. So with all this variation in the group, we can separate these different eye types into about 10 basic eye types. Where did all this variation come from, and why in this relatively small group of animals do we have this complete explosion of diversity of their eye shapes? So hyperid amphipods live exclusively in the midwater. It's actually in the definition or the diagnosis for this polyphyletic group. That polyphyletic issue is something for a little bit later. The midwater is the largest habitat on Earth. It's 99% of the living space on our planet. It's truly three-dimensional habitat. So we live in a three-dimensional habitat, but we're all tied to a two-dimensional interface, and so that limits the scope of our habitat. The animals that live in the midwater really are living in a three-dimensional habitat. Predators are coming at them from above, below, all the way around. It's a very different situation, and that explains a little bit about why we get these eyes looking in all different directions. The midwater is also highly structured in layers. One way to look at the layers is by the light, and since we're looking at adaptations to eyes, we want to look at that light variation. So in the top 200 meters, you get really variable light. There's variation in the color of the light, the direction it comes from, and the intensity of the light throughout the day and the year. From 200 meters to 1,000 meters, we call this the twilight zone or the mesopelagic, and there's very dim directional light. So basically, there's not enough light in the mesopelagic for it to bounce around and do anything, so it's all coming straight down from above. By the time you get down to 1,000 meters, there's no solar illumination left. There's no ambient light. But you do have light from bioluminescence, so there's still things to look at. So this kind of separates the midwater, this huge habitat, into three different light fields or light habitats that animals are living in. So we would expect three different light types, or three different eye types, or we would at least expect to find all those different eye types within just one of these habitats, but we don't. We find them all mixed up and all around together. We find close relatives with completely different eye types. So the goal of the project, this broader project, which a small piece of was funded, we're looking at the evolution of hyperid visual systems in order to better understand the drivers of diversification in the midwater. And we're taking a really multi-pronged approach to this, pulling in all the different, a bunch of different information about their biology to be able to understand where all these eye types come from. The project kicked off with Jamie's work for her postdoc, where she is a visual physiologist. She spent a lot of time in a dark room cryosectioning and measuring what wavelength of light animals can see, and then using histological measurements of their eye parts to actually model what they can see. So we can say, okay, Paraphonema gracilis, what does that species, what can it actually resolve, what can it see? When we link that information with something like what depth distribution does that species have, or what behaviors does it have, what animals is it feeding on, what animals is it associated with, that becomes really interesting. What we're doing with this project now is adding the genetic toolkit underlying all of this variation. So far, we've done a mini transcriptome that showed that retinal enrichment will work if we do a high-seq run. We recovered short contigs, but at least it showed us that all of the genes that we're looking for that are associated with eyes and vision we're getting those. So we should have a really nice data set soon. We went down, Bill Brown and I both went down to Baja, to the Gulf of California, and spent 17 days doing remotely operated vehicle in Midwater trawls. We collected for GGI tissue samples for 5 phyla, 38 families, and 54 genera. And then also for 7 transcriptomes from different species across this tree. 7 transcriptomes doesn't sound like much. These are really small animals, and their retina's even smaller. So it takes a whole bunch of them to be able to do a transcriptome. So with that, I would like to thank the funders for this project and SIBG-Biodiversity Genomics.
Thank you very much. Our next speaker, Alan Hare, is going to give a talk on genomic crosstalk, comparing the genomic interactions between a plant host, Theobroma cacao, and a fungal endophyte mutualist with those of a closely and distantly related pathogens. Thanks for coming up from Panama.
All right. You controlled the... So I'm going to tell you about fungal endophytes of plants. Fungal endophytes are in all plants you see anywhere. Most people think of fungi in plants in terms of pathogens, but there's no place you're going to see a healthy plant other than a growth chamber that is not chock full of fungi. And so these mutualists perform a lot of very useful enhancements for their host. Among other things, they can cut loss to pathogen damage. No, not the first. We're working backwards in time. So I get extra time, right?
You haven't
Okay.
started yet. Going back up to where you need to.
Okay, 2 titles. Ghosts in the Clockwork simply refers to the fact that all plants, as all animals, have got a huge microbiome associated with them. Generally speaking, people are only beginning to understand what the microbiomes do, but the fact is that without them, the host would not be the host that you come to think it is. And genomic crosstalk is what we're trying to do with our grant, which is to see what genetic signals the host sends to the microbes inside of it and what the microbes do in response and vice versa, hence crosstalk. Okay, so this is the summary of what I hope to be telling you. We've done a tremendous amount of work characterizing, you note the point mutation characterized, the effects of the dominant fungus, fungal endophyte, which is found inside of Theobroma cacao. It affects Theobroma photosynthesis, nitrogen metabolism, carbon allocation, stable isotopes of both of those. It affects photosynthesis, affects cell wall deposition, a huge slew of effects on the host if they have them versus if they don't. So we have begun comparing those effects which are produced fairly cryptically by the mutualistic endophyte with a closely related pathogen and with a distantly related pathogen. And as I speak, we are beginning to unravel what the different endophytes or pathogens produce, what gene products they produce inside the host that affects the host response to them. So this is the talk. How much time do I have left? Okay, we can go for it. All right, so all plants have got these things. As I've told you, this is a slide with the fungi stained inside of them. Roughly 2, perhaps up to 3 or 4% of the dry weight of a plant is going to be fungus, depending on the species. And the breakthrough that makes the work that I'm discussing possible was performed by Luis Mejia, my co-author on the project, he figured a way to, one, keep endophytes out of the plants, and two, put endophytes of our choice back in. So we have an experimental system where we can do this, produce plants that don't have them, and produce plants that not only have them, but have the ones that we want to test. So the general findings I've already told you, and the interesting question for us that we are going at with this project is, how is it that the endophytes that are inside of a host plant affect the host so that it is resistant to pathogens and herbivores? This is an experiment within plants. E minus, E plus means it either doesn't have the endophyte or does, and they're both challenged with pathogens, and the leaves that do have the endophyte in them are much more resistant to damage. So why does that happen? We initially thought that the fungi were probably producing chemicals inside the host plant, and so this was a bioprospecting project, but this is what we have learned now. The endophytes that are inside of a host elicit either up or down regulation of thousands of genes, let's say hundreds, consistently across different experiments. We can assign them to functions. This is a cartoon that shows how you do this. And so the general summary of those is here. The simple fact is that resistance and all of the other traits that I talked about have got a genetic basis, and those genetic bases in the host are affected by whether or not they have the endophyte and what the endophyte is. Okay, endophytes are closely related to pathogens. What is it that makes an endophyte a positive mutualist or a pathogen, a nasty thing that hurts its host? Previously, the endophyte that we determined is the most common endophyte in healthy cacao was mislabeled and lumped with lots of pathogens. They are close relatives. And so what makes them different? You run an experiment where you either treat it with the endophyte, which is called C. tropicale, a pathogen that's closely related to Theobromycula, or Phytophthora. And just visually, you see the heat map. This is a microarray. You can see that the pathogens look more similar. They look similar to the endophytes, and the controls that are not being treated are different. One of the ways that you can analyze the data is this. Use this kind of diagram. You find there are about 50 genes that are in common affected by the endophyte and the different pathogens. This is the ranking from highest to lowest of the upregulated genes by the most severe pathogen. This is putting the endophyte on top of it. What you can see is the endophyte, which produces health, has got a slightly different profile than the pathogen. We can compare all three, and if we do it, we can look at specific genes with specific products and why are each one different. What is it about the host? What is it about the pathogen or endophyte? That's the project.
A slight change of pace here. Rebecca Wong, who's here, works both at the Natural History Museum and with SCBI, is going to talk about her projects in collaboration with Intel and some of the experimental hardware that she's been working on for bioinformatics.
Thanks, Warren. So today I'd like to talk about the partnership that we're developing with Smithsonian Institute for Biodiversity Genomics and Intel, which is allowing us to optimize both for hardware and software biodiversity genomics and workflows. So the goal is to enable Smithsonian researchers to ask and answer genome-level questions. To do this, we're leveraging Smithsonian's strengths, collections, knowledge about diverse and rare species with cutting-edge genomics, and partnering with Intel, a global technology leader. So most genomics in the world is done on human genomes. So biodiversity genomics is challenging because we don't have, genomes are so diverse and we really don't know very much about most kinds of genomes that exist. So Intel is interested in these kinds of problems and we're interested in their help. So what we're doing is we're implementing and testing the performance of multiple algorithms and benchmarking real data sets on some pre-release Intel hardware that I'll tell you about. So what we would like to produce are optimized computational infrastructures so that as researchers are sequencing genomes across the institution, they'll know where to go and how to assemble those genomes quickly and efficiently, and developing protocols and pipelines for common tasks. This is a diagram that I made about a year ago, so it's probably changed a fair amount. This is pretty much a bare bones set of software tools that are needed by just about everyone doing biodiversity genomics in one way or another. Today, we'll focus on the top part, which is genome assembly. Genome assembly is a showstopper for all downstream analyses. And what I would first like to talk about is All-Pass-LG. That is the algorithm I'll talk about today, developed by the Broad Institute. That is a very highly regarded algorithm for assembling eukaryotic genomes. So what we've done is chosen some test data. So this is an African cichlid that was part of the Assemblathon 2 data set. Assemblathon was a contest among different institutions to see who could assemble the same data using different algorithms in different ways. And so we know what the product should be, what the assembly should look like. It has about a gigabase of a genome size, which is pretty average for if we average across all organisms, all eukaryotic organisms. In terms of raw data, it was over 500 gigabases. So we chose 3 data sets to test the performance, a small data set with 2 libraries. Obviously, we're not going to assemble the whole genome with this data set, but it's to have a rapid test for how fast the algorithm is performing on different sets of hardware. And then a medium set and a total data set. I'll talk about the small data set. These are the experimental computational infrastructures we used. The Intel machine, again, is pre-release. It has 72 cores and a terabyte of dedicated RAM. Nautilus, which is an Oak Ridge National Lab computer, has 4 terabytes of shared RAM and more than 1,000 cores. And then Hydra at OCIO has 512 gigabytes of shared RAM that we have for certain high memory nodes. So the interesting thing about the Intel machine is the only one of these 3 that has solid state disks. And so that is one thing that the Intel machine is bringing to the table here is very, very fast reading and writing when there are no moving parts. And so we wanted to test how that helps genome assembly. Obviously, the complication is that storage space on solid state disks is much more expensive. But if the trade-off in terms of speed is very positive, then data storage can happen somewhere else. So these are the results we have so far, just for a small data set. The Intel machine is performing really well, so completed in 4 hours. The Oak Ridge National Lab machine, 30 hours. This is probably due to the fact that their server is much older, but also the way it's set up, it requires every 24 hours a restart. So when we have a dedicated machine, we can see how important that can be for us. Hydra did pretty well, considering that it's not solid state disk drive. So again, this will help us in terms of the future of Hydra, determining what kind of nodes to buy and to increase our capacity to do genome assemblies. I'd like to thank the technical team at Intel. Sandeep Gupta and Matthew Taylor have been we have weekly phone calls where we talk about this. The next steps are to not only do 1 gigabase genomes, but what about some large plant genomes or amphibian genomes and maybe even some human genomes so that we can really test how fast we can get this going. At OCIO, the folks there have been really helpful in maintaining this Intel server, and that's pretty much it. Thanks.
Becca? Mike, are you here? Gates, yes. Come on up and get your talk going. Oh, there you go.
I thought I was number 5. 4 is missing today.
4 is missing, yeah. So, it should be down. Yeah. So Michael's going to give a talk on his work in the Southern Appalachian Mountains on genomic insect biodiversity.
Thank you. Actually, this project, as outlined in the title, hasn't happened yet. We leave at 3 a.m. tomorrow morning, a team of us to go down to North Carolina, but we have been to Argentina on our second funded GGI project. Both of these projects seek to sample as broad a spectrum of terrestrial arthropod diversity as possible to fulfill the goals of recollecting all life, even though we haven't collected all life yet. So I work with parasitic Hymenoptera, the smallest of which can be 100 microns in length. So our workflow necessitates the use of stereo microscopes. And in this case, we have carbon dioxide connected to a knockout block as used for Drosophila genetics work. And this works quite well for knocking down the parasitic Hymenoptera and other small flies, things like that. And it works really well until you run out of carbon dioxide, which is what happened in Argentina. So we had to come up with a different workflow. And one of the goals of these projects is to come up with best practices workflows that will allow others working with smaller arthropods to avoid some of the mistakes that we're learning about. So the first one involved duct tape. So we had a piece of duct tape, we folded it over on the edge so we could move it around. We would dump out ants and things like that, and they would slowly walk away and try to escape, but it gave us enough time to get an ID on them and then put them in the tubes for the liquid nitrogen. Of course, this ultimately didn't work as well as we had hoped. Here we made a modified water bath. There's a little bit of aluminum foil in there and ice in beneath the stage there. And that worked okay, but we ultimately wound up using just frozen ceramic tiles to slow things down. Our workflow was lined up like this. Here I am in the background working with some specimens. Matt Buffington is here entering the data as we're identifying things. And in this instance, we decided kind of at the last minute to take photos of all of our vouchers using our iPhones and a little lens kit I got on Amazon. Worked out quite nicely, and with the Bluetooth function for the iPhone, we're able to wirelessly transmit those images to the MacBook, then rename all of those files with the corresponding database number. And that really helped us troubleshoot a little bit later on when we had some missing labels. So these are some of the images. I mean, these are okay, and they're not publication quality by any means, but they allow us to put different sets of taxa into folders on Dropbox, and then we share those folders with the various experts locally or internationally. And we can also include things like, here's a stingless bee nest, here's a voucher for a particular host plant, that type of thing. So we found it to be quite useful, and we're going to be tweaking that a little bit and testing a different methodology in North Carolina. So our results for Argentina, we collected over 1,500 tubes across 3 classes, 14 orders, 66 families, and over 164 genera. And we imaged 372 vouchers plus associated images. This is for the Argentina project. So where are we going next? We're going to drive all the way down to Highlands Biological Station in the Southern Appalachians. This is a 23-acre research site. It has a pond, stream, a native plant garden. They do a lot of natural history work, especially with local universities, Western North Carolina among those. And then here is Highlands right here, and then there are a couple sites that we're going to investigate here with we're testing the mobile research lab, which is our SEL van. We'll see how well that works, but here is the Butt Creek serpentine outcrop with a very interesting flora. And then here is the standing Indian campground, and it has a lot of interesting bogs, seeps, and cove habitats that all have interesting flora. So the hope is they'll have interesting terrestrial arthropod diversity as well. Our goal here is 3,000 tubes. We're taking a total of 12 individuals, and we have coverage for 7 orders of insect plus arachnida. We're taking a documentarian who's going to be working on best practices videos and some photo documentation, and we'll use that and integrate into the GGI SI media outreach with Dustin Stonecipher. And one thing John Coddington is interested in is calculating specimen handling times across all these diverse taxa. So we're going to do a few different test sets for the different insect orders. Thank you.
Thank you, Matt. So you're going next?
What's that? Is it? Oh, Matt. There's Becker right there. Should be it. From there.
Okay. I'm Matt Becker. Is going to be giving a talk on the mechanisms of disease resistance to reduce the threat of chytrid fungus in Panamanian golden frogs.
Thank you. All right, everybody. So thank you for coming. So today I'll be talking about this lovely species, the Panamanian golden frog, and how it copes with a disease called chytridiomycosis. So this disease is caused by a fungal pathogen, Batrachochytrium dendrobatidis, and I'll just refer to as BD. This little guy infects the outer layers of the skin of many species and actually causes drastic population declines to extinctions of over 200 amphibian species. And that's worldwide. However, there are many species that can persist despite having an infection, such as the red-eyed tree frog right here, and then the cane toad here. And what I'm really interested in, what makes some of these individuals, populations, or species resistant to this disease. So I've been really interested in the bacteria on the skin and how these bacterial communities interact with this fungus. And a lot of these persisting species actually have antifungal bacteria on their skin, just showing that this bacteria can actually inhibit BD in the assay. Unfortunately, the golden frog doesn't have these protective bacteria or protective microbiota. And as a result of chytrid moving through its native habitat, they haven't been seen in the wild since 2009. However, we're really fortunate to have a very large captive population. And the main reason to have this captive population is to one day return it to its native habitat. Unfortunately, BD is still present in the environment, so we can't just put these guys back out there. So I'm really interested in using a probiotic approach where we can take these anti-BD or antifungal bacteria from persisting species and then treat golden frogs to prevent chytridiomycosis. So this is work in the mountain yellow-legged frog in California, as well as the redback salamander here in the eastern United States. So for my PhD work at Virginia Tech, I actually did a probiotic experiment with golden frogs, where I took a few probiotics, treated the golden frogs, and then exposed them to BD, and then had a comparison group where I just exposed frogs to BD. And this was a laboratory experiment. And as you can see from the survival curve, the probiotic treatments didn't work. So we didn't have really any differential survival from the BD control group. And the main reason for this is the probiotics didn't persist on the skin long enough to actually inhibit BD. But what is really unique and very cool from this study is that even in this BD-only group, we had 50% survival. So these guys are actually surviving infection, and this is really unique, as in 3 previous infection studies, only 1 frog out of over 200 actually has been able to survive infection. So we were wondering what's going on these frogs. So the first thing I looked at was infection intensity, so how many BD cells are actually on the skin. And in many of our survivors, we saw that they actually gain an infection and then over time clear this infection. So they're actually able to clear infection somehow. And this is in large contrast to those that died during the experiment. You can just see infection intensity just increasing until they die. So I kind of looked at the bacteria since I'm interested in the bacterial communities on the skin. And what I found is that these frogs that cleared infection had a unique community of bacteria on their skin. And in particular, there were 6 bacterial species that are in much higher relative abundance on these individuals that cleared infection than those that died of the chytrid. So this has led to some more questions I have. I want to really take this interesting correlation and see if we can get a causation and really figure out what the mechanisms of disease are. And that's what I'm doing here as a postdoc at the Smithsonian. So learning about whether or not was it the bacteria on the skin, or was it something about the host physiology. And if it is bacterial related, I'm really curious if we can take those probiotics and use those as a probiotic treatment for frogs, golden frogs, that do not have this protective microbiota. So to do that, I've been pre-screening captive golden frogs at the Maryland Zoo in Baltimore. We have over 200. And what I've been doing is characterizing their microbiomes, or just the bacteria on their skin. And based on our previous results, figuring out whether or not they have that protective microbiome, or potentially protective, and predicting them to survive. And then those that don't will be predicted to die. And then I will infect them with BD and see if my predictions are true. And then I'll also have a probiotic group under these predicted to die to see if I can change their outcome with these protective bacteria that I found, or potentially protective bacteria that I found in the previous study. So I'll be using 16S gene sequencing, as well as looking at the gene expression, hopefully of the bacteria, as well as the hosts, to see what specific genes are being differentially expressed between these two groups. So hopeful outcomes from this is that nail down what those defense mechanisms are in this species, so we can actually use that to mitigate the threat of chytridiomycosis, either by developing a probiotic method to prevent chytridiomycosis, or possibly establishing a captive population of resistant golden frogs that we may one day return to Panama. So with that, I'd like to thank a bunch of people, especially Brian and Rob that's here today, who are my advisors for my postdoc. And thank you all for coming. So thanks.
Thank you very much, Matt. Which is your talk here? There you go. Gotcha.
So you have to scroll up to the top side first. That one.
Okay, our next talk is from Vanessa Gonzalez, another example of how we are testing and seeing whether new technologies will fit, how they might fit into our research. Thank you.
Hi, today I'll be talking about Oxford nanopore sequencing technology, specifically the MinION miniaturized single molecule sensing system. So first, introduce you to the MinION sequencing system produced by Oxford nanopore. Here you can see this is the MinION sequencer. Attached to it is a flow cell that I'll go into a little bit later. This MinION sequencer is a little bit larger than a USB thumb drive, and it connects to a laptop, a portable device based on USB 3. And how this sensing system works, opposed to most sequencing systems we have now, which are called sequencing by synthesis usually, is that it's actually a sensing system. These nanopores are generated, are synthetically made, and are attached to a membrane. When the membrane gets an action potential distributed to it, it creates a current flowing through the pore. When a molecule passes through that pore, there's a characteristic current change specific to that molecule. And here you can see that certain molecules fit through this, basically 1 micron of diameter pore, and some molecules don't. But again, these current changes are specific to that molecule. And how that applies to DNA sequencing is something that Oxford nanopore refers to as strand sequencing. Here we see a double-stranded molecule generated by their specific library preparation software kits. And in the nanopore's case, they use this enzyme shown in green to help bring this double-strand DNA through their synthesized pore. This enzyme allows the double-stranded DNA to be unzipped and pass through that pore. And as in the previous slide, specifically to that molecule, there's a current change. There are specific current changes based on the nucleotides that are being read. These nucleotides, as opposed to sequencing by synthesis, which are generated 1 base pair at a time. Here, they can be read from 4 base pairs up to 11 base pairs at a time. This allows for long reads to be generated, up to 30 kb, which is 30,000 base pairs. When this sequence reaches the end of unzipping, the way that the library prep generates these double-stranded sequencing, there's a hairpin sequence here, allowing for both the sense and anti-sense sequences to be generated. Here at the Smithsonian, we've been awarded several units through the MinION Access Program sponsored by Oxford Nanopore. The MinION Access Program specifically has hundreds of participants worldwide, and what the function of this MinION Access Program is to allow for researchers to test specific and broad-range biological questions pertaining to this new sequencing technology. Here at Smithsonian, we've been awarded a total of 16 units, 9 here in-house, 2 at STRI, and 5 at the National Zoo. I'll specifically talking about the 9 units and our tests to assess the accuracy and output of these units. So currently, we've been involved in 2 phases of testing. For the first phase, all 9 units were employed. For the second phase, only 1 unit was employed, and I'll be talking about some empirical data based on the updated phase 2 tests. Specifically, what we are testing is both the hardware and the software, the hardware specifically being these flow cells. These flow cells have basically a 512 computer chip attached to them, which pores are now replacing each of those 512 ports. So there's 512 sequencing pores available on each of these flow cells. And here's just a sort of cartoon representation of the pore. And here is an actual protein model of the pore. And here, this diameter, looking down at it, is about 1 micron or a few microns wide. So what we have been testing is, are each of those 512 pores allotted to us per flow cell, are they active, are they functioning? And how well do they sequence? And what we're actually sequencing is a Heliobacter lambdaphage that is a known genome size and length, and we also have the reference genome. The sequence has already been obtained. So it's about 40 kb in length. And in order for us to sequence this and really test accuracy, we actually have to shear this 40 kb molecule down into smaller 9 kb pieces, and that'll just help us assess the output better, because generating 40 kb pieces is a bit difficult for any sequencer, and actually only one other long-range sequencer can probably output a sequence in that range, and that's the PacBio system. So specifically, the data I'm going to be talking about is testing this same lambdaphage sequencer using updated hardware, software, and library preparation systems. So in order for Oxford Nanopore to continue this MinION Access Program, they have been continually updating the chemistry, so the library prep system, as well as the hardware. That they always update the flow cells in order to, specifically for the flow cells, increase the amount of active pores, and that will in turn increase the amount of output generated using these tiny sequencers. So here, to familiarize yourself with the software used to control these MinION units, they specifically use a software called Minnow, and that is the control software. And here you can see all of the 512 pores represented as little dots. Each of the green dots is good. This is an active run, a snapshot of an active run. A green dot means that there is an actual sequence, a molecule being passed through that pore. All of the gray dots means that that pore is inactive for whatever reason. It could have not been active since day 1. It could just not have a DNA molecule passing through it at the time. This is a relatively good flow cell. This is actually a snapshot of phase 1 testing, because with the phase 2 testing, we had very few active pores, and that is a problem that has been sort of concurrent in other map unit users. And here, just present quickly some data. For the phase 2, we sequenced 2 flow cells here. And so basically what we generated was about 800 reads, and those reads were about 5,000 base pairs in length, with the largest one being 13,000 base pairs. And again, few flow cells were active, But what's promising is that we did identify about 24% sequencing error. And each of those sequencing, we recovered about 99.7% of the reference genome, which is very promising. And we recovered a 13,000 base pair piece that aligned to the reference genome with about 20% error. And I can go into it further if you have any specific questions as I'm out of time. And I would like to thank the MAP participants, GGI, and LAB, and the audience members.
Thank you, Vanessa. Ken. Here, you want to pull up your talk? Okay, our next speaker is Ken Rodak. He's going to talk about floral and biochemical specialization. Thank you.
So one of the fundamental questions in biology is how do species arise and persist? And in flowering plants, action is in the flower. So since Darwin's time, there's been this sort of general idea that specialized pollination increases reproductive isolation, which in turn increases speciation rates, which equates to more species. But recent models have suggested there's a lot more complexity to that equation, in particular how extinction plays into the part of it. So given this complexity, we want to ask, can genomics help? So part of the complexity is not only dealing with pollinators, there's pre-adaptation, there's constraints, and there's historical contingency. So in the plants that I specialize in, the Euphorbiaceae, there's a particularly interesting system that's been studied for the past couple of decades called Dallachampia, and I'll get into that in a minute. And in particular, I'm interested in how an inflorescence type such as that can turn into something like that through evolutionary time. So this is the array of plants that are lead up to the evolution of Dallachampia. So Dallachampia consists of about 120 species. Everything sort of related to it all has this other type of inflorescence structure. And another aspect of related to diversification is that when you gain some novel trait, such as in Dallachampia and this particular inflorescence, do you see an increase in speciation? So you can see that the things most closely related to Dallachampia, there are only a few species, so that suggests an increase in speciation rates. So what does Dallachampia have that makes it novel and perhaps have an increased speciation rate? It has this odd floral structure that has some advertising here for pollinators. And of particular interest is it has a gland here that secretes resin. And that resin attracts euglossine bees. And those bees come in very different species, and they serve as specialized pollinators. So different species of Dallachampia, they have different details in their structure that orient these bees, and the bees come and collect resin. And why would they come and collect resin? They use it for nest construction. So this resin is very unusual mixture of oxygenated triterpenes that doesn't dry, and the bees collect it. So while resin production appears to be the ancestral state within Dallachampia, there are the evolution of different types of reward systems within Dallachampia. So you can have a pollen reward system. The bees come and collect pollen. They've given up this resin gland because they don't need it. And you have this gland that secretes resin converted into a fragrance reward that's secreting, instead of resin, monoterpenes that the bees also collect and use as fragrances. So we have these reward systems. And if we sort of look at the sampling I'm proposing in this transcriptome project, we've covered all of these reward systems. This is how these reward systems in this schematic tree are related to each other. As I mentioned, the resin reward is ancestral. And then we have the gain of these additional types of reward systems. So that is the totality of the project. It's perspective.
Jesus.
I know, I hate it. I'm trying to keep it terribly simple.
Okay, our next talk is by Jesus Maldonado to talk about persinnities.
You at least have some data.
I'd like to start the talk by first acknowledging my collaborators, Dr. Chris Helgen, Dr. Klaus Peter Kefli, Roland Case, and Miriam Tsuchiya. My collaborators have been working on procyonids for the past 10 years and have tackled questions on phylogeography, phylogenetics, molecular systematics, taxonomy, and molecular ecology. And in spite of that, we feel that there's a tremendous amount of work that needs to be done to understand better this group of mammals. I also want to point out that Miriam Tsuchiya is a graduate student this is part of her dissertation project as well. But let's introduce the group of mammals that we're studying is the Procyonidae. This is a carnivore family that at first glance it appears to be a very ubiquitous family because it includes the raccoons, which are a temperate mammal that is everywhere, right? We see them in forests and even in urban environments. But there's also a diversity of mammal representatives in this group that are not so well studied. And a point to that is the second image, which is right here, which is the Olinguito, which is a species of mammal that was just recently described 2 years ago by Chris Helgen and his team. And we also have representatives of the coatis. This is an asonasua, the kinkajous, Porus flavus, the forest coati, Nasuela, and the ringtail cat. But in this project, what we sought to do was to sequence 2 representative genera, Potus flavus, which is the, oops, what did I do? This guy here and the ubiquitous raccoon. And the goals of the project are multifold. We sought to study the biogeography of this group, and the questions are when and where did this procyonid group diversify? We also want to tackle the taxonomy, and we are asking questions such as, does the current taxonomic classification of procyonids reflect patterns observed in light of the new revised morphological and genomic assessments from this study? We're also sequencing mitochondrial genomes, and we want to compare the similarities or differences between the inferences that are obtained comparing nuclear and mitochondrial markers. And finally, we're also very interested in looking at comparative genomics and evolution of adaptation in this group in the future. So once we have the genomes, we will be able to tackle these questions. And let me tell you why we think that this is an important group to study. If you look at this phylogenetic tree of the Caniforma, which is the suborder where the Procyonidae is included, which is the raccoons, the closest taxa that has been sequenced for full genomes is Amostella, the domestic ferret, and that's about 27.5 million years of evolution. So there's no representative procyonid that has been genome sequence to date. So in this project, we sought to, and we picked those two genera, Procyon and Pothos, because in an earlier study by Kefli and his colleagues, they had identified and finally, in 2007, resolved the phylogenetics of the procyonids, and excluding one genera, which is Nasuela. But this is the most complete phylogeny that we have based on 2 mitochondrial genes and 7 introns. And you can see that the Pothos is one of the most divergent of the representatives with 24 million years of evolutionary diversification versus the Procyon, which is a little more recent. And you can see that there's 2 species in Procyon that are about 5.5 million years diversity. So I think that what we want to do, as you can see, is this is just a tree that includes representatives from each genus. But what we want to do is look at the diversity and populate that tree by designing novel genomic methods that can extract DNA from museum specimens. And so we have the holdings, we have the specimens in museum collections, and we aim to design these protocols and tools to extract DNA from museum collections so that we can answer questions regarding systematics and taxonomy within each one of these branches. Part of the problem why Nasuela is not included in this tree was that the only Nasuela specimen we had was a museum specimen, and we weren't able to get the information from that. And so having and developing these tools is going to be crucial for us to develop a better knowledge of this group. And of course, comparing the behavioral and adaptations of these 2 very different mammals will also serve for future studies, looking at and comparing their genomic diversity. And with that, I'll end by thanking everybody. Thank you.
Thank you, Jesus. Our final talk, we have the pleasure of having Rebecca back to show that she's not just a technologist, but she's also going to talk about some biology, target enrichment data from flowering plants.
Hi. So this project, Vicki Funk is the PI. She's not here today, but I've been working on it for about 2 years. This is a project that has been ongoing, but the GGI award will allow us to expand the taxon sampling. So the family Composidae is one of the 3 major flowering plant families with around 25,000 species. It is about 45 million years old. And this phylogeny, which you'll see here, containing about 900 of the 1,700 total genera, has a poorly resolved base based on chloroplast loci and a couple of nuclear markers. So we were looking, so Vicki and colleagues were looking to expand that data set a number of years ago, and next generation sequencing, which is now, I guess, current generation sequencing, is allowing us the ability to do that. So in botany, genome level approaches are a little bit different than in other groups. The genomes are generally very difficult to assemble. They can be very large, they can be very polyploid, and even very closely related species can have different ploidy levels. And they can be really repetitive. So the sunflower genome, sunflower is a member of Composidae, has been sequenced. It is about 3 gigabases, which is about a human genome size, but it's 75% repetitive. But not only that, but those repeats are completely identical to one another, making the assembly really, really difficult. And so the only thing that could maybe overcome that would be an enormous amount of PacBio sequencing, which would be really expensive. And even then, there are likely to be very ultra-conserved elements, which are these areas of conservation that are generally in between genes, because the non-genic material in these genomes is generally just completely repetitive. So the options that we're left with are transcriptomes, doing phylogenies based on whole transcriptomes, which require fresh material from all taxa. And then what we're doing is exon capture, in which we're leveraging a few transcriptomes with museum collections and being able to sample from herbaria. This is a plate from our forthcoming paper showing some of the morphological diversity in composite. This is in addition to the very well-known species, sunflower, lettuce, artichoke, tarragon, and even there are some economically important taxa, such as the genus Artemisia, which has members with anti-malarial properties, and also wormwood, which is used to produce absinthe, is in that genus as well. So what I've been mostly involved in is the bioinformatics approach that we're using, in which we're looking to capture conserved orthologs. Conserved orthologs, but we're also looking to include duplicated genes with the hope that we'll be able to use the patterns of duplication across patterns of duplication across the family to understand the diversity both in morphology and in life history. And we have, a year ago, we had a workshop at the Botany 2014 meeting where we used the Amazon cloud to allow, I think we had 30 participants to be able to use our workflow on the cloud, which was a big success, and we hope to continue that in the future. So up till now, we have 23 species for 793 targeted genes. We're planning for 200. And this GGI award will allow us the funds to add 40 species in terms of sequencing. We already have those tissues in hand. And then also it will enable some field trips for Vicky to the Southwest US and in Peru to, we hope by the end of this year, to have 120 of the proposed 200 genera that we're looking for. This is our tree, just to show that the 23 species we chose as the initial starting data set span the entire tree of 25,000 species, but also are concentrated into interesting clades. So we could look at how well the loci perform, both in a closely related taxa and then across the backbone of the tree, and they do really well in resolving these relationships. So with that, I'd like to thank all of the collaborators and all the previous funding sources for this project. Thanks.
Thank you very much. A round of applause to all of the speakers for helping keep us on time. I'd like to thank also John Kress and John Coddington, representing the Smithsonian Institute for Biodiversity Genomics and the Global Genome Initiative. A reminder that June 17th, we are going to have a second round of these, and we hope that these will become commonplace going forward. Also like to point out, thank Lee Waite and Jessica Faison for their logistical help and getting this organized. And we don't have time for group questions, but hopefully the speakers will hang out here for a while and answer any of your questions. We have another event that's being scheduled, so therefore we're a little short on time. Thank you. Oh, and many of us will also be meeting over in the lunchroom or maybe spilled out into the public area. And that'll be another place we can reconvene and discuss some of these exciting projects and initiatives. Thank you very much.